of the physiological properties for those molecules. So we will try to do that also. Okay, so Alva Science. So this is found in 2018, and to be very much specific, this software is for QSAR and QSPR modeling, and it benefits researchers in various sectors and domains, including from drug discovery to pharmacology, cosmeceuticals, agriculture, material sciences. Of course, we are going to see that how it works and how each of the module for Alva Science works. So in Alva Science, we have total of 5 modules, but specifically the most important one are the 4 modules which might be visible on your screen. So the first one, that is Alva molecule, we will see in detail. That is for data curation. We have Alva Desk. Alva Desk primarily used for the descriptor generation. Then we have Alva Model for model building and validation. And then we have Alva Runner for model deployment. Deployment means specifically if you're having a novel data set and you wanted to check the activity of those novel compounds against the generated model or the created model, uh, with the help of the training data set which you are like having with the biological activity, then you can go for Alva Runner. So amongst this, we will be seeing in a little bit detail, starting from the Alva molecule. And yeah, if you people might be having any doubt in between, or if you want me to repeat anything, you can stop me as I've told earlier, and you can ask it. Yeah. Sorry, I think my
Yes,
screen
sure.
is stuck. Yeah. So starting from Alva molecule. So Alva molecule, as I've told it to you, it is for the data curation and worksheet for visualizing the molecules. So if you might be having a data set, and that data set might be having 100, 10,000, 1 lakh compound, you can easily visualize those compounds. You can see the properties, and which might be already present. If you might have downloaded that data set using some sort of a PubChem or a drug bank or binding DB or Chembl database, then you can see the properties and all those specific which might came along while you download those particular one. When we talk about the data molecule curation, you have, like, of course, you can primarily go for multiple structure corrections if there might be charges, isotope, no carbon, aromaticity, if there might be radical atoms which might be present in your data set. So all those corrections you can do. And of course, you can locate that molecules over the PubChem and Google uh, uh, Chem services. I would be showing this functionality also while we will be jumping on it. Let's start from here. So just focus from here. It is comparatively useful for you guys. So first we will go for the standardization of the data set. So standardization means, suppose if you have downloaded the data set and that particular data set might be having missing hydrogen atoms, or there might be something what we call as free radicals which might be present, which you don't need. There might be charge on the atoms, or you wanted to or there might be some excess valency which might be there in the data set or the few compounds. So we are standardizing the compounds, or we are correcting the basic structure so that we can generate the descriptors for those one. So that is basically the standardization of the data set, which starts from here. Then we have the duplicate analysis. Duplicate means, suppose if you have generated or if you have a data set of 1,000 compounds, and if there are duplicate compounds. So as you can see on my right hand side, there is one graphical user interface which has been provided, and you can see multiple molecules are there which are like duplicate of each other. So all those duplicate molecules will be assembled or will be shown in a single color. So this particular compound is duplicate of this. This is the duplicate of this. So we need to delete such sort of compound which might be duplicate of each other. So in that way, what you can do, you can easily remove that particular duplicate compounds and keep one of them And of course, there are certain criteria or restriction you can put while managing the duplicate. And of course, you can use it for the further process. Then you have the scaffold analysis. So scaffold analysis basically uses this Bemis-Markov framework. So it specifically divides the molecule into three parts. First is linkers. Secondly, the aromatic rings and the last one is the side chains. So specifically in your data set, how many number of molecules are having a similar scaffold? So suppose this benzene ring is there, and as you can see, this is scaffold number 2. And this is like 453 molecules are having this particular benzene ring. So you can go for the scaffold analysis also, and you can generate the Bemis-Markov framework for each of the molecule, and you will get that how many number of molecules are having the same scaffold. So just take for an example, if I have if I'm working on some sort of psychedelic drugs or some sort of serotonin analogs, we know there is a very common ring that is indole ring. So you can see that the scaffold indole might be there, and it is like almost in all the molecules. So you can see that most of the scaffold, the indole scaffold, almost in all the molecules, it will be shown up here that how many number of molecules have that same scaffold. Then we can go for edit, sorting, and filtering the data. So it's very much simple. Of course, you can go for filtering, sorting, and editing the chart. You can generate few of the graphs. You can generate the histograms on the basis of the molecular weight and the other functionalities and the properties which you are calculating. So here, one second. Yeah. So specifically, this tool is not for generation of molecular descriptor. For molecular descriptor generation, we have another tool or another module named as Alva. But of course, we can go for calculation of approximately 88 physiological properties combined with molecular descriptors. And those might be topological polar surface area or log P or Lipinski rule of 5. So all those specific physiological properties can be easily calculated using the Alva molecule. So this is there. So primarily we are going to see the Alva molecule. Then we will move on to the Alva disk. So this is how the graphical user interface of Alva molecule looks like. And of course, if you're operating it on Windows, you can do so. If you can work on Linux also, if you have Mac also, then this software is suitable for Mac operating system also. So all three are fine with that. If you wanted to know more about the Alva molecule and wanted to read in depth about this particular software, so what you can do, you can go to the help section, and there is this Alva molecule help, and you have certainly like how the graphical user interface works. So as I've told it to you, of course we will be doing it now, but just if you wanted to get an insight, so every single thing is mentioned up here, how you can generate the graph, then how you can load the molecules, how you can filter and sort and edit the molecules, then how you can go for the scaffold analysis, how you can go for the duplicate analysis, how like what's the theory behind it, then what sort of standardization it is basically present in this software. So every single thing you have like in very much of depth in this help section, and of course the type of physiological properties it can calculate and the descriptors. So all those drug likeness indices, ring descriptors. So you can come up here and you can see that what all it can calculate. So it will be there along with the theory that which might be useful when you go to publish it, and you can understand it more about the physiological properties which could be calculated. So I'll just close it and go back to the window. So now let's open a set of molecules here. So we have this case study, which I already took it from the Alva Science website. Yeah, one more thing I just wanted to show it to you. If you will go to the website, and of course, you have the credentials with you, that is the user ID and the password. If you will go to the resources and go to model studies, you will get very many studies here for your practices. For your practices means if you'll go to the learn more option, there is already the data set along with the project file is there. So you can download it from here and you can just practice it by yourself. So every single thing which has been there and what's the result of the process and everything has been defined up here, starting from Alva molecule, then calculation of the descriptors and then Alva model, and then after that deployment of the model. So you can visit those case study and read it, and it will make your concept more clear. So once we'll go, I have already done one of the case study, which I will be showing it to you. So, yeah. So once we will come up here, we have this data set. So we have this data set which we will be using. So let's, of course, you can, if you will come up here, you can see that how many number of formats it support. If it support the .stf format, it supports smiles format, it supports small format, and almost all the format which you can which might be you are using in any other drug discovery software or any other open source. So you can use those one. So I will be opening up this data set. Yeah. So this is a particular data set. In the downside, you can see the number of molecules here. In this particular data set, we have 1,303 molecules. This is one of the view for which we will go to the extreme downside. So you can see all the molecules which are present in this particular data set. If I'll come to the view and wanted to change the visualization part, of course, I can do so. I can go to the molecular worksheet. So of course, the few of the things like log BCF value or the training and all those basic things has been defined up here. So in the view section, you can define it using different visualization like grid section, worksheet section, or you can go for the column selection also. So those things are very easily you can go through. Then if you wanted the molecule to be represented with a molecular name. So as you can see here, the name has not been defined, but if in your data set, the name might be there. So the name will be visualized here. So you can see all the names. Of course, for this data set, we don't have name, only numerical ID has been given. So those ID we can see, but of course not the full name, because we might not be having the name on the on the the drug bank or from where we have downloaded it. Okay, so
Yeah,
once we,
so here I
yeah,
have a question. So,
yeah,
which
yeah.
are the common sites from which we will get these data sets?
Ma'am, there are many like where you can download the data set. Like, of course, you can go from PubChem is there. Uh,
PubChem, I know. What else?
Drug bank you can use. Binding DB is one of them. Then there is Campbell. Then there is a Pass server. Pass server is basically you can use it if you are dealing with the natural compounds. So there are ample amount of, there is zinc database. So ample amount of database, and it totally depends like what sort of studies you are going through. Like or what sort of data set you want. Suppose if you want if you're working on a specific target and working on some sort of anti-neoplastic activity against a specific target, then you should look for the structures which might be having the biological activity, or you can also use some other activities for generating the data, such as binding or IC50 value, or any other value like so of course these are few of them which you can rely on.
Yeah, so if that can be provided to us, just the link of those sites and the
Sure
list.
ma'am, sure ma'am, I'll just write it down. Okay, link of the data sets, sure ma'am.
Yeah, thank you.
Okay, so once this has been done, so now we can also come to the view section and let's try to explore more. So of course, if you want different representation of the hydrogen atom, I'll just change the view section so that we will be more focused on the molecules. So of course, these molecules are there, and if we will go to the hydrogen section, we can see on hetero or terminal atoms if we wanted to represent the carbon and hydrogen, so that could also be possible. And if I want some other representation on terminal atoms, so that also can be possible. So I can come up here, or on the heteroatoms if I want the hydrogens, or if I want to show all the hydrogens, so that can also be possible. So different view strategy can be implemented here, and you can see the molecules in different. So I'm just off so for the clear representation. Then we have, if we will go to the highlight, of course, we can change the color, the background color in white and black, but of course, then we have these substructures, so which I was talking about, the Bemis-Morpho features. So when we'll go to this Bemis-Morpho features, it basically divides the whole molecules in a ring, linker, as well as the side chain. So as you can see, each of the molecules has been done, and of course, we can see the linker molecule, we can see the side chain, and we can see the rings. So this is one of the technique, or we can see this is one of the function in this particular software for a representation of the molecule. Now if I want to, if I don't wanted to go for the Bemis-Morpho approach, and I wanted to just go for like substructure searching, so I wanted to search all the structure which might be having the benzene in their in their structure as a substructure. So what I can do, I can just give the smarts format. Okay, so once I'll go, sorry. So we can give this is like, of course. So here we will see that we have to check the smart structure for this particular benzene, and of course we can go for the Bemis-Morpho approach, which might have developed here. So if I want a single molecule to be visualized in a whole window, I can select that particular molecule. So suppose if I want this molecule to be enlarged, and I wanted to see, I can go to the single molecule mode, and that can also be done. But right now we don't want that. So this is few of the viewing option, and we can see those viewing option in here. And of course, we can now there is aromaticity. So what sort of, if there are, we can see the aromatic structures in like various aromatic rings in all the structure. What sort of representation we want those in the Kekulé form, or in the aromatic form, or in their original form. So that can also be changed. So these are different sort of visualization options, or to change the structures. Now we will be moving on to the standardization of the data set. That is one of the most important one. But before going that, we will check the structure. So once we will check the structure, it will check it for, I'll just try to maximize the screen a little bit so that the people can see this. Yeah. So we have checkers for like multiple structure. If there is unusual valence atoms, covalent ionic bonds might be there, or if there might be radical atoms, if there might be presence of some non-standard atoms. If you wanted to uncheck it, of course you can do it, but it is of course a checker. It will give you the number of molecules for each of the one. So if I'll just click OK, so as you can see, the bond molecules are 158 according to the criteria which has been defined. Multiple structures, we have 0. Unusual valence atoms, there were like 25. Total charge, like the atoms which we have the charge on, there might be nitrogens which have the which might be carrying the positive charge, or so those are also defined. Then we have the non-standard atoms, or the molecules which are containing those. So we have 24. Of course, aromaticity, how many number of molecules, and a radical atom. So if we will come down, so we can see there is aliphatic compounds also, long straight chain compounds also, and we can see that if we will come extreme downside, this is the data set. Now we will go to the structure section and go to something like standardization. So what it is going to standardize it. So of course we have seen what sort of loopholes are there in the data set while checking the check structure option. But I am going to go for all sort of standardization for now. So how you can do, suppose if you wanted to go for any specific standardization, so add charge to quaternary nitrogen, of course we can do that. We can remove the exceeding hydrogens. We can add the missing hydrogens. We can go to standardize the nitro groups. We can also go for removing the radicals or the free radicals which might be present. We can neutralize the atom if they might be having some charge, neutralize the full molecule, or of course these things we can do, and retain the biggest fragment, that is okay. We can clear the isotope, and of course we can press OK. So now my data set has been standardized. So in my data set now, there is no free radicals. Of course, there might be presence of some duplicate compound because we haven't started with the duplicate analysis, but it is standardized in on the criteria what we have just did it. Removing all those free radicals, adding hydrogens to the missing ones, or satisfying the valency. So all those things has been done. Now, once I'll go to the tool, so I can go for the property calculation, but first we will go for the duplicate analysis. So once we will go to the duplicate analysis, you can go by column also when we will go to the molecular grid section. But right now I'll go for the default and go OK. So it says in my data set, there is no duplicate found. If there might be duplicate, then what you can do, you can come to the structures and tools, and you can go to the manage duplicates. So automatically the compounds which are duplicate of each other, it will be deleted from your data set. Or you can go to the show duplicates. It will show you all the compounds which are duplicate of each other. So that is there. So now once we have did, now the most important thing, I'll change the view to the grid, and I'll go to the tool option and calculate the properties. So now all the properties which we have already told, like starting from Lipinski rule, TPSA, and of course, if we will come to the last section, we can see few of the properties like psychotic 50, depressant activity. All those things has also been defined that whether this particular molecule are having this sort of property or not, related to depressant, related to psychotic, and different one. Lipinski rule and oral bioabsorbability. So all those you can go through it and you can check it. Then you can go for the charts option. You can go to generating the histogram scatter plot for you. So suppose if you wanted to generate it for the on the basis of molecular weight. So you can see all those molecules which are having a specific weight. So it will tell you that the number of molecules which are having a specified weight. So of course you can go through it, and the other property you can also screen it. So the molecules which are having the neoplastic activity. So that can also be done. So of course you can segregate it like that. And this functionality we will be also having in the Alva desk. So all, if you wanted to explore more, you can go to the Alva molecule help section as I've told it to you, and you can check it more about it. And this is the basic like what we can do within the Alva molecule. So now I will stop for a bit and ask if you people might be having any sort of doubt, you can ask.
Yeah, I have one query. The
Yeah.
interpretation, once we get the results,
Mhm.
so the interpretation is also given somewhere of each parameter.
Ma'am, yeah, interpretation is like, for example, let me show it to you how we are going to interpret it. For let's go to the worksheet part.
Properties, yeah, this one, suppose.
Okay, so suppose interpretation is like 1 and 0. So specifically, it's like the binary digits which has been given. So 0 stands for like specifically it is not carrying that particular activity, and 1 stands for it is showing the positive for that particular activity. So that is something how you can go for. And for all these value, of course, we can go for like, of course, log p value or the partition coefficient value. So we have to look for it that what we want. We want it like to be the molecule, the more lipophilic in nature, or we want it more hydrophilic in nature. So accordingly, we can see the value like for the log p value, or for the other hydrogen bond acceptors, hydrogen bond donors. So it totally depends on the structure or the studies what we are doing. So suppose if I'm doing studies for CNS, I want compound to be more lipophilic to cross the blood-brain barrier, for an example. So I will look for the activity. Okay, how much should be it in the range that the log P should be there. So accordingly, I'm going to segregate the molecules out. Of course, we can do that. So that is an easy task. So if you wanted to segregate the compound, so what you can do, suppose the M log P value has been given. So you can filter it and you can create the value here. So suppose I want I want the compounds which are having the log P value equal to suppose 1 or 2. So I just can type it here and I can press OK. So there is no compound which is having the value of log P 1. But of course, if there might be in your data set, it will pop up here. So like that, you can segregate it and you can interpret the data.
Yeah, because I was more keen on this physical chemical properties, like Lipinski rule of 5. So again, can you just show me how it is showing here?
Yeah, ma'am.
The data.
Sure, ma'am. So I just, yeah. So in this particular one, of course, we can see, suppose if specific properties we wanted to calculate, as you were like asking about Lipinski rule. So we have to search, like Lipinski rule consists of what? Of course, the molecular weight is there, log P is there, hydrogen bond acceptors are there, donors are there. So on the basis of that, we have to go one by one and search the criteria whether it's following or not.
Yeah, so the criteria, if the general criteria are there, can it be shared with us?
Sure. I will share that particular general criteria if it might be there. Cause there is like specifically there is no column which directly say. Of course, it consists of 4, 5 proper. So I'll share that criteria with you for sure,
Yeah,
ma'am.
so details of this, because this will be a core work in pharmacology.
Okay.
So for these
Yeah, ma'am.
parameters.
For if you wanted to know more about it, of course, it is already it is there. Like if you'll go to the help section, I'll just show it to you. If I'll go to the properties, so we have all these indices, constitutional, ring descriptor, suppose molecular properties are there. So every single thing has been written up here. Like suppose the TPSA is there. So all things and the values or TPSA quantity, like everything has been defined up here that what it actually tells and what is the interpretation of it. Yeah, so of course, but if other than that also, if you might be having any doubts, so and the references has also been produced. For example, drug likeness one as we were like discussing right now. So all those things has been given in the help section. So the hydrophobicity and but of course, if you want, I will share the list also and it will be more helpful. So I will share the list and all those properties, what it actually tells. So there are, as I told, there are approximately 88 descriptors and physiological properties are there. So all those properties are listed here.
I have one question.
Yes, sir.
If suppose I want to make a new structure, or maybe
Mhm.
some other functional groups are there which I just want to remove and put one more functional group. So will it be possible in this tool?
Sir, editing won't be like possible. Of course, we can go for copying the smiles and pasting it, but of course, like drawing the structures will not be possible.
Okay.
Yeah.
So if
Uh,
I'm putting the smile string,
mhm. Yeah, it will work. If you will put the smile strings, of course, it will work. So if you'll
oh, I
go to
can
the
draw
smile,
the okay, I can draw the structure in some other tool which
Mhm.
I would like to just visualize. I'll put
Yeah.
that smile string into this tool. So will that work?
Yes, sir, it will work. If you will put it in the smiles format, it will work. And of course, if you wanted to copy smiles from here also, that would also work. And if you will save the molecule in STF format, then there is no need of like drawing the smiles, then taking, you can save the molecule in any format like STF, smile, small 2, and then you can open it here.
Because sometimes what happens, certain functional groups are not properly binding. So we need to modify those functional groups to add something new. And we have to
Yeah.
check out whether that new functional group is going to be conformationally stable with the rest of the functional parts of the structures. So they
Yeah,
should not
yes.
clamp to the other part. So if that is there, then that would be really fine.
Yeah, sir. Of course, you can do so. And suppose if there are like multiple also, of course, for the each and every we usually go for property calculation when we have like, of course, some molecules. So when you will go to here, you can go to the open molecule section. And of course, right now I'm just closing. So here we have Smiles format, we have MDL, we have like of course you can go for the STF. So all those format are easily you can open it from here. So if you use some other molecular sketcher like Chem Sketch or something like that, so you can draw those molecules. Even you can have the whole library. You can save that whole data set or library, and in a single time you can open all those molecules in this graphical user interface.
Okay. Okay.
Yes, yes, sir. Okay, so this is about the Alva molecule. Now I wanted to save this data set. So how I am going to go so I will go to the save as option. And of course, if I wanted to save it in the Smiles format or SDF format, I can do so. But I've already saved this, so I'm not going to save it again. So here this data set already exists, so I'm not going. But of course, if you wanted to save, you can save it as suppose data set underscore try, and you can save it. And now it will be saved, and you can open it also in the Excel format. And of course, you can use the results in for your research purpose and all. So this is the first step what we are going to do in the Alva molecule section. And ma'am, I have noted it down that if you want more information about the properties and all, so I will just try to share some papers and some information regarding that. And of course, you can go through it. And if you might be having any doubt, you can reach out to
Yes,
me.
that would be very helpful. Huh? Thank you.
So let's move on to the next one. So I will be moving back to the slide section. Okay, and please let me know if my screen is visible. Yeah. So
Yes.
now we will be moving on to the Alva disk. So Alva disk specifically for the molecular fingerprint and descriptor generation. As I told it to you, this full suite revolves around QSAR and QSPR modeling. And of course, so the most or the one of the most important step is like generation of the descriptors on molecular fingerprints. So this Alva disk will be used for that. Once we have standardized our molecular data sheet or the data set, now we have to generate the descriptors or the fingerprints for it, so that that could be used for generation of the or creation of the model or building the model. Talking about the Alva disk, Alva disk, we have approximately 6,000 molecular descriptors, and of course consisting of 2D and 3D molecular descriptors. What all descriptors it has, it is very wide range. So ranging from descriptors for calculation of molecular properties, we have descriptor 2D, 3D constitution, like ample amount of descriptors are there. And of course, we can also calculate the descriptors for 3D molecules. And within the graphical user interface, we can convert the molecules from 2D to 3D, and it uses the universal force field for it, so that it runs in such a way that the coordinates would be correct, and of course the structure would be optimized. So here we can go for the property calculation as we can do so, but in the Alva molecule, it is like a small what we can go for only 88. Here there are more than of course or approximately 6,000 you can go for. So starting from TPSA, log P, log S, and what all we can do within the Alva disk, we are going to see now. What are molecular fingerprints? We can go for calculation of the hashed and non-hashed max 166 keys. and other molecular fingerprints. So basically in molecular fingerprints, in a very basic sense, if I'll say, it is like a binary representation of whole of the structure containing 0 and 1. And it specifically tells us about the what all substructures which are present within that particular structure, how it is connected. So all those things will be defined within that particular molecular fingerprints. So binary representation of a normal 3D or 2D descriptors. So we can also go for structural pattern representation. And now we will be moving on to the Alva disk. So I'll close this as I've already saved the project, and I'll open the Alva disk for calculation of the molecular descriptor. So now we can open the molecules, we can open the project also. So if I'll go to open the project, and I have saved that. So here we have. So we have these descriptors which I have saved using that. And of course, if I want, I can also use the data set from. Yeah. So this is my data set. So I've standardized this data set. I have calculated the properties also. So I can visualize all those properties here also. So as you can see, all those properties are easily visualized here, and we can calculate so many different models. Now I can change the view. View section is approximately same here. If you want the descriptor list, you can also see the list of the descriptors. What all descriptors are actually being calculated and what all we can do for calculation. So if I just zoom it a little bit. Yeah. So as you can see, the list of the descriptors are already there. And as I've told it to you, if we'll come to the downside, there is approximately 6,000 descriptors consisting of 2D and 3D. So I'll close it. And if you want to know more about it, you can go to the view help section and can know more about each of the descriptor. But here we will go for the calculation of those descriptor. So we have analysis part and chart, and of course we can go for, but let's go for generation. Yeah. One more thing, if you wanted to generate, so all these molecules are 2D. Now, if you wanted to go for generation of the 3D descriptors also, primarily you need to convert those 2D molecules to 3D molecules. So as I've told it to you, from here, this graphical user interface, you can directly convert those molecules. But here I'm not doing so. Here we will be working on the 2D only. So let's go for the descriptor calculation, and we are going to exclude all the 3D. Why? As you know that we are not involving the 3D one. So all the 3D descriptors has been removed. Now these constitutional indices, ring descriptors, 2D atoms, 2D descriptors, charge descriptor. If you will come up here, you will see all the sub descriptors which are present under those. So we can do that. Now we can present, just press OK. And you can see downside that the calculation for all those descriptors has been started. So yeah, so we have generated all the descriptors for or all the desired 2D descriptors or the desired 2D descriptors. And as you can see that it is there with the molecular weight. So till now, do you have any doubt? Of course, if you want to go for more calculation, we can go for something as structural patterns. So something, if you will go for the structural patterns, means if that particular molecules are having same. Suppose if I wanted to add, I'll add something as support. We have to add the smarts here. Smarts like the representation of that particular one. Now I wanted to see the benzene, all the structures which contains benzene, it is same as that. And I can name the molecule as A or something like that. So those can also be segregated using those, but right now we are not going to do that. If I want each of the molecule to be separated in the left hand side, so you can see the visual representation of those molecules. So basically this tool is for fingerprint as well as the descriptor generation, and the specialty that it provides almost 6,000 descriptors which can be used. So till now, I just wanted to ask if you people might be having any doubt regarding the theoretical as well as the practical thing which I've shown. Uh, then we can save the project. Yeah.
I think it's clear to me.
Okay. So after the descriptor generation, yeah, anyone wants to ask anything?
No, no, that's fine.
Okay. Then we can go and we can of course save those descriptors. If we want, we can save the project. We can save the fingerprints also. But right now we will save the whole project as it is. So as you can see, the project which has been there, I have already saved it. So I'm not going to save it again. So this is one of the study. So once we save this, now it is ready to be opened in the Alva model. So Alva model, I'll just close this and show you. Let's move on to the Alva model part. Yeah. So this is to create or build the QSAR or QSPR model. And of course, we need a data set. We have standardized the data set. We have generated the descriptor for those data set. Now the time has come that we will build a model using that particular data set along with the descriptor. So as you can see, there is a figure. So it explains everything on its own. So this is Alva model. We need to give the alvadesk.prg means the descriptor for the structure what we have generated, or the fingerprints what we have generated along with the target value. So what is this target value? This target value can be any biological activity which you might be interested in or which you have the practical results for. It could be IC50 value, LD50 value, or EC50 value, or something like docking score. That can also be a target value for the list of the structures what you have given. So using those two things, we are going to generate the model. Then we are going to give that model to the Alva runner. And what? Then this Alva runner, then if we might be having some sort of new molecules what we have, and we wanted to predict the activity, binding affinity, or any other property, we can use the Alva runner for that. So is there any doubt? Shall I explain it again or is it clear? Like this figure actually explains everything. You will get the predicted target or the predicted activity for the novel compounds for the new set of compounds. So is there any doubt or shall I explain it again? Like, have you got it? How it works?
Yeah, if you can just repeat it again.
Yeah, sure. So we have Alva Desk. So in Alva Desk, we have generated the structures. Sorry, Alva Desk, we have generated the descriptors. So we have descriptors for the structures, for all the structures which we have standardized and all those things. Now we need the target values also. So those target values can be any biological assay values. It could be IC50 value or EC50 value or some other. It could be values such as binding score or docking score and all those things. So those structure along with their activity, along with their biological activity values, we have to supply both those things to the Alva model. Now, this Alva model using those two things will generate a model. Now, this model we will supply to Alva runner. And Alva runner, now when we supplied the model, if we are having new set of compounds that can be any novel compounds which we have synthesized in the lab, or that can be compounds which we have designed using computational chemistry. When we will supply those compounds to the Alva runner, automatically it is going to predict the value on the basis of the earlier trained model. Have you got it?
Yes, yeah,
Okay.
now a little better. Only thing,
Okay.
like you said, the target, huh? Target could be IC50 value, or it could also be some enzymes which are involved in disease processes.
Yeah, like enzyme inhibition, like that you are saying, like
Yes.
Yeah, it could be any, like if even this target value can be docking score also. Even this target value can be EC50 value also. But the thing is, if you are having the practical values or the wet lab experimental value, then probability of getting or probability of generating a perfect model is is increased to a certain amount. Because as you are giving the wet lab results, or and specifically what I would say now, you will get most of this IC50 values using the databases like Campbell, or a few times these IC50 values for many of the data sets are also available on drug bank or binding DB. So they also provide, like if you have data set, of course the biological activity value has to be provided. So this is the one, and we will be seeing it. Uh, so we can go for the feature selection using the genetic algorithm. We will be seeing that genetic algorithm during the automatic model generation. So during the automatic model generation, it uses the genetic algorithm. So we will be doing those things. And of course, it has got the regression and classification model. Under the regression and classification model, what all algorithms, what all machine learning algorithms, KNN it uses, support vector. So all those things we are going to see it now. So let's move on to the practical part of it, and we will open the Alva model. Yeah. So this is how the graphical user interface of Alva model. And I know that many of the students might not be familiar about those algorithms. So they might not be familiar about the support vector algorithm or something like KNN algorithm. So what you can do if you wanted to read more about it, so you can go to the help section, view help, go to models, and of course everything has been defined up here. So there are different distances what we are going to see, though the understanding of all those things, of course few of them are required, like what is R square, or few of the other values like what is basics of it might be required while you will be using this software. Of course not too much in detail, but if you are knowing it, it's good. So you can know from here, and you can try water support vector. So under the classification or the regression model, so regression, what is the regression model, classification model, what it is, how when we use the classification model, when the, so specifically classification model, for example, if it is used when you don't have the continuous value, or your data is segregated, or you have the binary number 0 and 1, whether it is showing activity or not showing activity, whether your compound is toxic or not showing or not toxic. And when you have the continuous result, or the data or the quantitative one, then of course you are using the regression model for that. So what sort of regression model it is there, and provided by the Alva. So what is this k nearest neighbor? So if you will go up here, you will find a small text regarding that. So how it actually works, so that you can just learn a little bit about it. How did support vector machine actually it acts? So all those things you can just read it a little bit. And of course, if you might be having any doubt in it, you have to read about it. I would be very happy to clear it. We can have the session also, and recently we will be visiting, like within 1 month or something, in Mumbai also. So there also we can have session, and till then you can get little bit accustomed to the software, read theoretical part, then we can have one session for that also. Okay, so let's go to the GUI interface, and let's go to the file section. Now we have already generated the descriptor. We will go to the Alva file, and let's go to the desktop, and we have this. We will open that. So it will take some time, as the number of molecules are like 1,303 molecules what we are using for generation or creation of the QSAR model. So this is the master data set. So as here you can see all the molecules are visualized. They have the ID, and of course the value or the activity what we will be using is this one. So this is there. Then we have the training values, and all those things are actually given up here. Now we are going to import. Of course, we have done. the external variables. So these external variables are the values. You can give the BCF data set or just and BMF also. This one is, yeah, can open it. Yeah. So as you can see, the log BMF value has been given for all of the one. So what we can do for right now, we can directly open the project. So as I've done, so we have directly opened the project, or you can supply the value as well as the external variable one by one. But as we don't need it, so we can now we have to segregate or we have to split the model into test and training data set. So how we are going to do so? So we will go to the model. We will go to like the data set, split training and test data set. So once we come up here, so we have like type of splitting what we wanted to do and the training size, how much we wanted to keep. But I'll go by column or by size also you can go. So let's go for the training and the value has already been defined. That is one. So what is this one value? So basically 1 and 0 has been defined for each of these compounds and we are going to split it on the basis of it. So all the molecules which are having the value of 1 for the training will be in the one segregated data set and the one which are having the 0 value will be splitted into the test data set. So I'll just press OK. So as you can see, the training one all has the one value and downside you can see there is 978 molecules which are which will be used for training the model. And we have of course the test data set. So this is like 0 for all the molecules which are having the value 0. Now we are going to generate the model. You can go to the model. You can also go for the automatic model generation, which uses the genetic algorithm. But for right now, we are not using that. First, we will go to add model. Then we will also see with the automatic model generation. Let's go to the add model. As I've told it to you, we are not going for the classification model because the values here, as you can see, are like continuous value or these are the quantitative one or these are not segregated into. So we will go for the regression model. Go for next. We will be using linear regression, the simplest one. Let's go for the next one. In training, we will define the training. In the test data set, we can define the test data set. Press next. Now the target we have to define. So our target is log BCF. So what is this target? We have to define the biological activity. So here the biological activity, what I'm considering is log BCF value. So I'll come up here. Now I can select few of the descriptors which I wanted to use for the generation of the model. So how am I going to select those descriptors? So as I am actually doing or iterating the same uh, case study which has been already done. So I will be using those one only. So if I'll go to learn more. And as you can see, these are the model descriptors which has been used. So what I can do, it's very easy. If I'll come up here, I can just copy paste those descriptors which are used. I'll go to next. I'll go to select. Just copy X1A. This is one of the descriptor. Because anyways, we are not going to use all the descriptors. Of course, we can use that, but here it's not necessary. Then we will go for some other. Suppose we will go for C1089. Okay. So are you getting like we are actually selecting the descriptors which we need for the generation of the model? If you have doubt, do you have any doubt why I'm doing so or something like that? Okay, then we will go for the mass.
No, we'll go.
Okay, okay. Then we will go for selection of some other descriptors like.
Is the PDF file available for this or is only online?
No, PDF file is also available. And online you can go, of course, you can search the paper by its DUI, and I think so it will be available. And the whole project is actually summarized here. So if you wanted to read, whole project is summarized here, what they have done, what's the interpretation and every single thing.
Oh.
Then if you wanted to select the other descriptors, you can go. Yeah, so we can go for this one. So total 6 descriptors they have used and M log P value. Yeah, so we have this M log P value, and if we wanted to keep, so just press OK. So total 5 descriptors we have selected. I think one more is left which we can use. 5, I think there is one more descriptor which is left. I think this one. Okay, yeah. Yeah, so now total 6 descriptors which has been used, and of course, if we want, we can actually go for selecting the fingerprints also if we wanted to use the fingerprints for the generation of the model. But here I'm going through the descriptors and I will press next. So let it be it exclude columns if the missing values are found. So missing values means if there might be columns in which the activity might not be defined. Okay, and if there might be data which is required to generate the models are not there, so we can exclude those columns. And of course, there are few of the feature reduction technique which is there. So let it be on the default and finish it. So as you can see, now the model has been generated. I'll just press it a little bit down. You can see the values. You can see for the training data set. You can see the values for the test data set. You can see the values for the test and training both data set. Now we have generated the model, and now the model is ready so that we can use it over the novel compounds, newly synthesized compounds, newly generated computationally newly generated compound, so that we can apply this model and find out whether they are having some relevant activity or not against a specific target or against a specific biological action it might be having or not. So this I have generated using the model generation one. If you wanted to go for the automatic model generation, that also you can use. So what you can do, you can go for the automatic model generation. Now we know it is a regression model. It uses the genetic algorithm. Of course, we can give the R square or some other criteria if you wanted to use. Let it be R square. We are going to use KNN. Okay, and we can use Euclidean distance. Training and test is okay. We can select the descriptors. If you wanted to go for selection of the descriptors, let's select uh, let's select, but not all external. Okay, so it's like 4,215 descriptors what we have selected while generation of the automatic model generation, and press OK. And then we can press next and let it be in the default and press finish. So now you will see it will go for generation of the automatic model, and it will find. So now the best score, as you can see, it is 0.722. so the more the score closer to 1, the better our model would be. So it will found like it is going to screen amongst 10,000 models using the descriptors what we have done. So automatic model generation you can also use for your model generating or creating the QSAR or QSPR one. So now you can see the best one what we have found is 0.755. Of course, it is going to take some time as it is 10,000. So I'm going to pause it for now. And if you will pause it also, you will find the best one, what they have used it, and what the best model what we have got according to the value for the R square. So till here, is it clear? Like we have in a very short summary, if I will tell you, we have standardized the data set using the Alva molecule. We have generated the descriptors or the fingerprints using the Alva disk. after generation of the descriptors, we use those descriptors along with the structures to create or generate a model. So for generation of the model, you can go manually by selecting the model. Of course, you can go for the KNN, or you have to select whether it's regression or classification, or you can also go for the automatic model generation. And then you will get it, and you can save that model also. So suppose if I wanted to save that model, I can save it here and I can open that model. I will get. So here now it's come to the interpretation part, which is a very important one. So this is the actual value. This is the predicted value. And this is the residual. Residual means how much is the difference between the actual and the predicted value, or how much correction it is like needed between the two values. So as you can see now, we haven't even generated the 10,000 models. Within the initial 50-60 models itself, we have predicted the value which is very close to the actual value. So once you are going to screen it for the larger data set, then you will see that the values you are going to get for the model, it is far more accurate. Okay, so is it clear like what it is there and how it works, what is residual, what is predicted? Because now we will be moving on to the next one, that is Alva runner. Any doubt is there, so you can ask freely. Yeah.
No, it's
Yeah, I
fine.
think this requires a lot of practice, but yeah,
Yeah. Not an
theoretically it
issue.
is clear.
Yeah,
Theoretically
ma'am,
it is fine.
yeah. Yeah, ma'am, you can, of course, you can practice it and I will be available. So you can ask. And of
Sure.
course, I've told it to you. If you want, the link is already there. You can log in, and if you want, I will share the models, or so on that models the students can practice. And of course, I will share the links for the research articles also, which has been published using this. So while reading that, they will get more clarity about it.
I can just want to go through that PDF file. Can you share the PDF file?
Yeah, sure, sir, sure, sir. I will
Good.
share the PDF file.
Even I.
So yeah, I will share the PDF file along with the links. So almost so many, and it used to get updated, I think. So once some new paper has been published, so it will get updated. And of course, I will share, sir, for sure. I'll just write it down. Yeah. Okay. So now we have generated the model. I'll take a 1 minute break. I'll just drink water and then we can.
Yeah, sure.
Hello. Yeah. Now we can move on to the next one. That is the last, that is Alva runner. So first we will try to understand it again. So now we know we have generated the model. We have every single thing. We have saved it. So we have saved the model, that is alvarunner.project file. Now these are the new molecules. So new molecules for which we wanted to calculate the physiological properties. It could be any environmental properties, biological properties, or binding up, or any other like properties. If you have the new molecules for which you wanted to go for, just we have to feed those molecules along with the Alva runner project. Then it is going to predict the target, or the predict the values for these targets, or the target values. So as you can see, you can apply the model. So it is very simple. We will be seeing it in the live phase also while the demo. So these are our molecules. And of course, these are the predicted values. So these are all the predicted values which have been predicted using the model what we have used. So this is KNN model, as you can see, and it is tells the prediction fold, like the specific molecules fold, like which specifically lies outside the applicability domain. And there is all the other, if we are generating multiple models, we can do it and calculate the predicted values for the multiple models too. So that we can do. Let's see this. So once we come up here, we can save this project. So how you can do so? It's very simple. You can export the models. You can save the project. You can just go for save project as, and you can save the specific project. So I've already saved it. So I'm not saving it, but it's very simple. You can save it from here. Once everything is saved and everything is done, so automatic model generation is, if you wanted to restart, yeah, one more important thing. As you people know that I've already stopped it while it generated only like 82, out of which this one, I can restart it from here. So automatically it will be restarted from the last model and it will again start to search the best model. Again, the process would be started. So as you can see right now, the R square value, the best which it has found is 771. So of course we can move more, and the more it will be searching, the probability of getting a better model will be more. So of course you can do that. So we can pause it, and of course if we want, we can save this model also. Just save it. So we have three models now. This, we have second model, we have third model, and if we want, we can come to the model one, we can change descriptors. There are a few other options, and of course we can save the results. Now we will be moving on to the last, that is Alva runner. Yeah, so this is how the graphical user interface, it is nothing different from the interface of the other modules. If you will go to the help section and go to the view help section, of course you can operate this Alva runner within the command line interface also. And as you can see, the values, the predicted values are there for the new compounds. So that would be there. And there is applicability domain. So what is this applicability domain? So whatsoever compounds which actually falls in that particular applicability domains will be given a number one, and the 0 1 will be highlighted. So if there might be one which doesn't falls into that applicability domain, will be given a value, or it will be represented in a binary style like 0 and 1. And of course the 0 will be one which is not falls under that specific applicability domain. Then if we'll go to the model, of course we can go for the regression classification. It is all same as the last one. So I'll just move on to the GUI. So how we are going to do that? First we have to open the molecules. So let's open the molecule. We will go to the same, that is desktop, and so we have those molecule, that is so we have this data set, and or we can use from BMF also. Just a second. We save data set and model values. So we have this in the text format and we can use those. Yeah, we can have those values also. So it's in the smiles format. If you want the values to be there. So I'll just export it like this. Sorry, I think so. This is not the correct. Yeah, this is the one. Actually, we have saved the file. So data set. Uh, yeah. This is the data set. Sorry for inconvenience. Actually, I was confused that which is the novel compounds for which we have to predict the values. So this is a novel set of compounds, and for this we don't have any values. Now we are going to use the model which we have generated using the Alva model to predict the values for these molecules. Okay, so now how we are going to do so? So I have imported all the molecules. Now I have to import the values. So now the model. So it's very simple. You can go for the open project or recent molecules. You can go for the open project. Now we know the project, it is there. This is the project what we have done. Yeah, you can see the R square value which is there and the RMSE value which is there. And there is one more what we have done. So multiple projects were there here. Yeah. So as you can see, there were three models what we have generated in the last section, if you can remember. So for the one model, the R square value is 0.657. For the second model, which we used using the KNN, K nearest neighbor, so there is the value is 0.729. And there is this 0.750, which is one of the best what we have seen. You can see the other details also. So I'll press OK. So for all the models, it has created or it has given the value. So right now we have only done for the first one. So as you can see, the values has been given at the predicted values. So this prediction fold, the specific molecule falls outside the applicability domain. So how much it falls outside the applicability domain, it has been shown up here. So as you can see, the value all is approximately 1. And there is this AD distance, which tells us about the applicability domain, if it might be 0. So as you can see, for no of the compound, the value is 0, according to the model what we have generated. So I think it might be clear to you that we have to give the novel compounds, and then we can calculate not only the applicability domains, but how much the specific molecules falls outside the applicability domain according to the values. So is it clear?
Yes.
It's clear.
So
Can we
this
bind it by 230? Like, have